what are the applications in the plants? First is the golden rice. Golden rice is getting from the biofortifications which was developed by the Ingo Potrysis and Peter Beer. So here we take the 1 beta carotene which is borrowed from the daffodils gene, and then we take a vector which is the TI plasmid, and then we'll produce a product that was the recombinant DNA, which is a mixture of both of this. And then with the gene gun method we'll introduce this recombinant DNA into the plant, and then we'll get the product that is the Oryza sativa. And this Oryza sativa is basically a golden rice. So golden rice which is very rich in a vitamins A, and it helps to deal with the diseases like prevent night blindness and nyctalpia and solve this problem. Second, in the application in the biotechnology, that is the hirudin, which is the anticoagulant in nature. So we take one hirudin gene which is from the hirudo nadicinalis, and which is basically from the leech. In leech salivary gland there is a presence of hirudin. So we take the hirudin gene and then vector which is the TI plasmid, and then with the TI plasmid we have recombinant DNA which is the recombinant DNA will get, and with the help of gene gun method will introduce this gene into the another plant species, and then we'll get the product that is a Brassica rapus. And this Brassica rapus is very with the castor oil and hirudin will get. So we want this separate castor oil, separate hirudin. So this can be separate with the help of the downstream processing, and then we'll get separate castor oil, separate will get hirudin, and hirudin which is the anticoagulant. Third application which is given in the NCERT will basically cover all the NCERT applications from the biotechnology, which is important for the exam point of view. So next is the herbicide resistant plants. So what are the we have main crop. So what is the actually thing is that with the main crop we have herbivoids also. So we want that herbivoids to stop growing, so we'll use this method that is the herbicide resistant plant. So in those plants we have cyclic pathways, and cyclic pathway there is a production of the EPSP synthesis. So what we'll do, we'll get the amino acid which is a tryptophan, and with the help of EPSP synthesis we'll try to produce the auxin which is responsible for the plant growth. So there is a glyphosate which is the inhibitor of the EPSP synthesis enzymes, and it will help to block the EPSP enzyme pathway. So remember, glycophosate, it's not glycophosphate, it is glycophosate which is the inhibitor, and it will inhibit the synthesis or the production of auxin with the help of the EPSP enzyme block pathway. And in the biotechnology we'll use EPSP enzyme overexpression of the main crop. So this is the method of the herbicide resistant plant. Next is the insecticidal plant. Insecticidal plants we have Bacillus thuringiensis, and in Bacillus thuringiensis we have protoxin is found in this particular enzyme, and in protoxin we have a gene which is called the chrysin, and in chrysin we have cryprotein, but cryproteins are inactive form, and in the insect we want in the it will go inside the insect's gut, and the pH is alkaline, so it will get there active in the pH gut of the insect. This cryprotein will become active and then attach to the midgut, and this will lead to the proliferation of the wall of the insect, and the insect will not be able to digest or eat the food, and insect will die because of starvation, and insect die. So this is very helpful. And bt cotton, which is the first GMO, that is genetically modified organized plant introduced in the India. So this is the first GMO genetically modified introduced in the India, that is the bt cotton. So the next thing is the RNA interference or the gene silencing. This is the three name of this RNAi. So what will is the first thing, we know that we have DNA with the then will form RNA, and then RNA will be responsible for the formation of protein. But if the RNA will not form, then there will be no production of the proteins. So first, this is the tell by the Andrew Fire and Craig Mello. or which is they've done experiment on C elegans. And in India we've done, basically the experiment is done in the tobacco. So what happened, we'll find that in some point of time there is the yield of the tobacco is decreasing. So we have to find the reason that why the yield of the tobacco is decreasing. Because M. incognitia, this kind of a bacteria who's responsible for the that he take all the nutrients from the dicot plants, and he always take in that nutrition. So because of that our yield in the tobacco is decreasing. So in the eukaryotic cell we have the natural defense mechanism. This is a process, actually you can see. So what will we do, this is the cell, and this is the nucleus, and we have DNA already present, and this is the double stranded DNA is introduced. Basically this is the TI plasmid. So when we introduce the double stranded DNA, we'll see the sense and antisense strand, and we break this double stranded DNA into the parts, and this is called the siRNA, that is the interference and the protein. And this is called the RISC complex, RNA introduced silencing complex, which is the expression gene. The next one is the human insulin, which is called the humulin. In this is developed in the 1983 by Eli Lilly company. Humulin they produced. So what they did, they took the chromosome 11 beta cells of the pancreas, we know islets of the Langerhans, which is called, they produce insulin. Always remember, beta cells of pancreas cells they produce insulin, but it is a proinsulin. So we have proinsulin A, B, C, and we have to remove the chain C from this proinsulin. So we remove insulin. Insulin is the functional now protein, this will do the function of the protein anabolism. This is the hypoglycemic, promote the adiposis. And now we'll take the gene for insulin, we'll take the vector which is the PBR322, then recombinant DNA will produce, and this is will take the E. coli, then we'll do the downstream processing, and then we'll get the product which is the humulin. And this is the A chain, this is the B chain, and they are attached together by the disulfide linkages, and this will humulin will function, this will help to prevent the diabetes mellitus. So this is the all about the applications of the biotechnology.